Well, good afternoon. I will just continue from where Nick has left. So I basically take care of the MS Biobank. And this biobank is basically not a very similar to a blood biobank, which you have normally heard of. This is pretty much a research-based biobank. And the idea of the biobank is to use samples, human samples, samples from people with MS, and try to study MS per se using human samples. So this is something, a very recent development which is developed in biomedical science in the last 7 to 10 years. And that is what we want to try and use and move away from animals and guinea pigs, which are normally done in basic lab research, and move more closer to humans and try to see what actually happens in humans, and then try to extrapolate or work from there to make better understanding of the disease and go forward from there and see what we can do. So the whole biobank basically is divided into there it goes. Okay, so the biobank has two parts. One part is recruiting participants, and now what we are recruiting here is slightly different than what Nick is doing for the family study. We are happy to recruit even individuals in the family who have MS, whereas Nick, as he said, that it has to be 2 or 3 for being a part of the family study. But we are not discriminating that part. We are just taking everybody in. We are also inviting people who are non-affected within the family, or anybody else who is also willing to come and donate, who has no family history of MS. We are welcoming them also to make a complete set of population of people living in a certain area. That's how we look for it. So this is the first part of the biobank. Now, in the biobank, as per today, we have more than 100 participants of MS people who belong to all the 3 types of MS: primary progressive, relapsing remitting, and secondary progressive. From these 100 participants, we have already created 50 stem cell lines on which Kelleen and our group is actually working to understand and study how MS goes ahead and add some new insights into MS. I will talk about this part in the second phase, how we do that. Within the bank, we have mainly 3 types of cells. One cell, basically the first ones are which we directly collect from participants whom we recruit, that is the blood cells. And the second and the third one, which is induced pluripotent stem cells and the laboratory generated brain cells. Now this is a little different. These 2 are made in the laboratory. It has nothing to do with participants as such, but these are the main precursor for the second phase, which is studying MS. So that's the part which is more interesting for us, which is making iPSCs and making brain cells, which basically help us to study MS. So I'm sure everybody here in the audience have heard about what stem cells are, here or there in news about regenerative medicine, stem cell therapies and everything, but what exactly are stem cells? So stem cells are basically very immature cells, and what gives them this specific ability is that they can divide forever. They are immortal in a way. And also they can differentiate or convert into different cell types. So that gives it a very, very good viability to work and study downstream cell types or different cells of the body if you want to do that. Now, the question which comes to everybody's, which I've been hearing always, is are all stem cells same? No, all stem cells are not same. You would have heard of bone marrow stem cells or umbilical cord blood stem cells, which people nowadays preserve for the next generation and kids and for better life. But what we are looking is for induced pluripotent stem cells, or the last line, which is iPSCs. Now, these cells are basically cells which mimic an embryo, basically in a way that when the life was actually formed, when you or me were as an embryo, a single cell. So we are basically talking about science fiction movie. If you are, anybody is interested in science fiction movie and has seen movie, we are talking about not back to the future, but back to the past. So we are going back in time, taking an adult cell from a human being that is collecting your blood or anybody's blood, and pushing them back to the stage when you were an embryo. And from there, we start working forward and trying to make brain cells, which is basically a replica of how your brain cells are behaving. So the more the number of participants we have, the more different kind of brain cells we have, which will basically depict me, X, Y, and Z person in the brain. So that gives us a very good population study-based analysis of how different brain cells work. Now, the advantage what we get is people having MS and people not having MS, especially in families, where we can specifically identify what is different in brain cells of people with MS, or what is different with in people who have not got MS, in this case of families. So that is a very strong potential. And this is just a schematic. What I wanted to show you what we do in the lab this is a picture of, this is a picture of the blood cells what we collect from individuals, and this is under a microscope. If you put them under a microscope, our blood cells pretty much look like this. Now, on these blood cells, we gave a cocktail of magic factors, and these magic factors basically transform them into cells. By 25 days of transformation, what we end up is a cell or a group of cells which basically mimic your embryo. So basically, you put them back in time from a mature cell back into a fetal state or an underdeveloped state. Now, what we plan to do in this whole thing is use these cells, that is the stem cells. So we start with the population, recruit families or individuals, make stem cells, and then with these stem cells, we can make various kind of brain cells, which we are looking at, maybe neurons, astrocyte, glial cells. And of course, it will help us in studying genetics from brain cells, as what Nick is talking about. As well as we can also try drugs, which we can never give to a human being to be tested, because we can't use humans as guinea pigs, but we can always use cells as guinea pigs and try whatever we want to do them. And it gives a direct correlation of how the drugs will behave when later on we move them into clinical trials and return some better therapy or better things to people with MS, and that probably would be something which we are looking forward in the next coming years. With that, I would like to thank my supervisor and with Kelly, with whom we are doing this work, Professor Bruce and other members of the community who have come forward and been very happy to participate and donate blood. If anybody here is interested, please get in touch with me. There are flyers in your bag, and you can get in touch with me on later time also. And of course, none of this would be possible with the fundings and every support from others. Thank you very much for your time.